Hi there, everyone. This is Hasib, and I'm with Cyrus and Jane. We are the Humid Fund, and today we are showing you guys our research into the effects of humidity on catnip growth. So before we got into the experiment, we decided to ask a few questions that would help us lead to an answer that we were satisfied with. First off, how does a change in humidity affect catnip over the course of its growth? And then more specifically, will the xylem area, stomatal conductance, and stomatal density of catnip vary in different levels of humidity?
We predicted that the catnip would show a decrease in xylem area, stomatal density, and stomatal conductance when they are grown in wetter conditions than their counterparts that are in less humid conditions and in the control condition. For instance, the stomatal density in the leaves would likely be smaller when they're grown in conditions that are more humid than others.
We based our study on 3 different humidity levels: humid, arid, and control. These humidity levels were regulated by a cover to ensure each group had its own atmosphere, consistent amounts of soil, light levels, and groundwater provision. This cover was made with a wire shelf made into a cube that was then wrapped in plastic wrap. The 2 covers were placed on our humid and arid groups to maintain their humidity levels. To further maintain the humidity levels inside the cover, a cup of water was placed inside our humid group, and a cup of desiccant was placed inside our arid group. With the environment of our groups in place, we aim to measure stomatal density, stomatal conductance, and the area of our xylem.
As part of the process for this experiment, one of our methods was to take stems from our plants and create cross-sections out of them, and then take them down to the lab and put them under a microscope and do some measurements of area. And we didn't only do xylem area, but we also did size of the whole stem to look at stem growth between different groups. And we also looked at the area of the xylem as a proportion to the stem. So we could see whether or not arid or humid environments would create larger xylem bundles, smaller xylem bundles, or maybe no change between them. For leaf measurement, we ended up snipping the second leaves from the top so growth wasn't too new or too old, and we put them on a plate so that we could order them by group and by plant number.
We gathered 3 leaves from each of the 3 humidity groups. A layer of clear nail polish was added to the underside of each leaf sample. The stomatal peel from the leaf is then collected and examined under the microscope. We compared the stomatal density between each of the 3 groups.
The third method used was a perometer. After calibrating the perometer for the greenhouse conditions, all catnip samples in each of the 3 groups had their leaves measured by the perometer. The device calculated the level of stomatal conductance, or gas exchange in leaves, as well as showing how changing initial environmental conditions could affect the conductance rate. From the perometer data gathered, the control and humid groups generally exhibited a similar range of conductance rates to each other. In contrast, the arid group often had significantly higher rates in their leaves. The arid samples also generally had less issues with leaves drying or browning than either of the humid or control groups. Barring a small number of outliers, the 3 groups generally followed a similar trend in conductance rate with reasonable averages. There were significant differences in conductance results across different days, which is explained by varying weather and time of day. Overcast afternoon versus clear morning, which could not be avoided or mitigated due to logistical constraints.
No significant differences were observed between the stomatal peel of the catnip leaves. Our methods for gathering the stomatal peel on the catnip leaves were inconclusive.
So here's a quick overview of our table for the cross-section measurements. You can see we did some averages. We included proportion of the xylem area, not just to the stem as a whole, but also the vascular bundle of the stem. And then if you look at the bottom row, we also did some other measurements on xylem vessel area specifically, and we picked out 5 specific vessels in each stem, and we measured their area and then averaged that out for each plant and then compared those averages to each other as well. Here are some pictures of our cross sections that we chose to put up for you guys. The first one that you can see is a stem cross section under a 5 times magnification, and then the second one under a 40 times magnification. In that one you can see the individual xylem that we chose to measure for the average xylem area as part of the leaf measurements. For the results of the cross-section measurements, we actually noticed that stem area was noticeably larger among the arid plant group compared to the control and humid groups. There's actually an 8 to 15% increase in area respectively from the arid plants compared to the other 2. But for xylem area, there was no difference in straight up area measurements. But because there was no difference, we found that arid plants had a reduced proportional area. So they took up 8 to 12% less space in the total stem area compared to the other two groups. As for the difference in area of each individual xylem vessel, we found that arid plants had smaller vessels in terms of area. There was about a 10 to 20% reduction in average area from the arid plants compared to the control and humid group.
Arid environments found to be most conductive for effective catnip cultivation. CO2 conductance of arid plants found to be 2 to 6 times of plants in control and more humid environments. Arid environment led to thicker stems with thinner and proportional smaller xylem. Ways to improve experimental design. How do you think we can improve in our design?